Hi everyone, this is Silvia de Santis. I am Principal Investigator at the University of the Institute of Neuroscience of Alicante. Today I'm going to talk within the subject of alteration of the cannabinoid system in animal experimentation and clinical pathology. My specific topic today is going to be methods for detecting changes in the endocannabinoid system, neuroimaging methods like PET, SPECT and MRI. Let's start with a brief scheme of the course. I will start with talking about PET, positron emission tomography. I will first describe a little bit the physical principle governing this technique. I will then talk a little bit about the radio tracers, and then I will present some of the main and recent application of this methodology to the study of the endocannabinoid system. Afterwards, I will explain single photon emission tomography, SPECT, the physical principles, and again the main applications. And then we will move to MRI, magnetic resonance imaging, the physical principle behind the technique. I will talk about the different kind of contrast that can be used in an MRI experiment, functional, structural, spectroscopy, etc. And then I will describe as well some of the main applications to the investigation of the endocannabinoid system. And eventually I will talk a little bit about the comparison between these three methods and try to talk about pros and cons of each of them and compare them. Okay, so let's start with the first part of the class. I'm going to talk about the PET, positron emission tomography, but first of all, a very, very brief introduction on the endocannabinoid system. The endocannabinoid system is a biological system composed by the endocannabinoids, which are endogenous lipid-based retrograde neurotransmitters, their receptors, and their enzymes, which are responsible for the synthesis and degradation of the endocannabinoids. This is a scheme of all the molecular targets involved in the system. And what is important to stress is the endocannabinoid system plays a crucial role in regulating various physiological processes, including mood, appetite, pain sensation, inflammation, and immune response, but is also involved in several pathological processes. This is from this picture on the right is from a recent review in which both involvement of the cannabinoid system in physiological and pathological processes is highlighted. The author especially put the attention in the context of physiological processes about the reward network, the attention, the emotional response, impulse regulation, memory, and associative and working memory. But also, as far as pathology, it has been involved in several of them, ADHD, depression, addiction, OCD, schizophrenia, and so on. For a complete list, I remind you to this very nice recent review, the two recent reviews. Okay, so let's start with the first technique. It's positron emission tomography, or PET. Let's start with a general overview about which is the target of PET and how does it work, and then we will go a little bit into details. PET is a molecular imaging technique that employs positron emitting radio tracers to visualize and quantify biological processes in it. The PET imaging of the endocannabinoid system is based on radioligands that selectively bind to cannabinoid receptors, which are the receptor 1 and 2, we will see it a little bit later, or to the enzymes responsible for their degradation. How is it done? By labeling these radioligands with positron emitter isotopes, the distribution and binding affinity of the cannabinoid receptor or enzyme can be mapped. So in this case, a tracer, we will see it a little bit in detail, but just as a general idea, the tracer, in this case is one based on fluorine-19, is composed by a part that binds to the target, the receptor or the enzymes, and the second part, which is a radioisotope, which is emitting positrons. This emission is what is captured by the PET camera and by capturing this, we can access to distribution and binding affinity of the target of interest. This is an example of one of the very first PET images of the cannabinoid receptor 1 tracer A in the panel A, and a blocker in the panel B in a monkey brain. So, what is the physical principle behind the positron emission tomography? A positron is a subatomic particle which has the same mass as electrons but has a positive charge, and it belongs to the antimatter. The positron is by its nature unstable. Actually, when it comes into proximity with an electron, they experience an attractive electrostatic force, which is due to the fact that they have opposite charges. As they approach each other, the positron and the electron undergo an interaction which is called as an electron-positron annihilation. What is annihilation? During annihilation, they merge and the combined energy is released into two gamma rays, which are high energy photons, moving, this is very important, they are moving in opposite direction. Each one carries a very specific energy of 511 kiloelectron-volt, equivalent to the rest mass energy of an electron-positron for the conservation of energy. Just to note that this annihilation process occurs very rapidly, typically within a few picoseconds. Picosecond is 10 to the minus 12 seconds. Okay, so let's see a little bit the experimental flow behind the positron emission tomography. As I said before, the tracer is composed by two parts, a biological active molecule and a positron emitting radionuclide. Examples are fluorine 18, carbon 11, oxygen 15. The combination of the two makes the PET tracer. The PET tracer is injected in the body, is produced locally, close to the PET camera, and then it's injected in the body by means of an injection. And once in the body, the tracer binds to the target, depending on which specific tracer has been chosen. It binds to the target, the receptor, the enzyme, etc. So, since the tracer contains a positron emitting radionuclide, very quickly the tracer undergoes radioactive decay emitting a positron. This positron travels a very short distance, normally a few millimeters, before colliding with an electron and undergoes this annihilation process, generating gamma ray. And this is captured by the PET camera, which is a camera specifically built to detect these events. How does it work? Just a few details about the PET scanner. It's composed by a ring-shaped detector which surrounds the patient or the animal, consisting of multiple scintillation crystals coupled to photomultipliers tubes. The gamma rays interact with the crystal, releasing light photons, which are then converted into electrical signals. And what is important is that the signals are assigned to a specific location, so to a specific event, under the principle of coincidence detection. And this is because these gamma photons that are released have are released in opposite directions. So by looking for coincidence events in the ring, one can detect where the event came from and generate an image. But what is an image? A PET image? The computer, by analyzing these coincident events, reconstructs a 3D tracer distribution image. And normally, the maps that are shown in a PET experiment, that are produced in a PET experiment, represent the standardized uptake value, or SUV, which is calculated by dividing the tissue radioactivity concentration by the injected dose and normalized to the patient body weight. And this is an example of a paper of a few years ago with a map of the standardized uptake value, and in this case it was for the distribution of cannabinoid receptors 1. Okay, let's go now a little bit deeper into the targets of the PET imaging in the specific for the endocannabinoid system. We say that the targets are mostly receptors or enzymes. Probably the most accessible one are the cannabinoid receptors 1, especially, but also 2. The cannabinoid receptor 1 is primarily found in the central nervous system. The receptor 2 is located predominantly in the peripheral tissues and but also in the activated immune cells. But recently has been also shown that it is also present in the healthy brain. And another target for PET image is the fatty acid amide hydrolase, or FAAH, which is an enzyme which is involved in the breakdown of anandamide, and it's very important for the endocannabinoid system, and is also can be also targeted by a PET tracer. And lastly, the fourth target is the monoacylglycerol lipase, MAGL, which is another enzyme which is involved in the degradation of 2-AG especially. All four of them are targets of the PET, but the most used one is for sure the CB1 receptor CB1. What do we need? In this slide, I collected a few desirable characteristics for the PET tracer. So I hope so far you have understood the importance of the PET tracer in the process beside the camera. So of course, a whole field of research is developing new and effective PET tracers. Which are the desirable characteristics of a PET tracer? So first of all, it has to have high specificity to the desired molecular target or physiological process, minimizing non-specific binding to unrelated tissue or structure for obvious reason. It's also ideal if we can have high sensitivity, because we want to detect low concentration or changes in the target. We also would like to have a favorable pharmacokinetics, including rapid and efficient uptake into the target tissue and organ, appropriate distribution, minimal metabolism or excretion before imaging. We also want rapid clearance from non-target tissue through, for example, renal excretion or hepatic metabolism, because in this way we can enable the visualization of the target signal with minimal interference from surrounding tissues. We also would like to have optimal radiolabel decay, an half-life of the decay that balance imaging time requirements and radiation exposure, so minimizing the radiation exposure for the patient. We want also the tracer to be able to be synthesized easily, and we want it to be stable. As I said, there is a slide on this, but the tracer is normally synthesized or in a site which is very close to the PET camera, because immediately it has to be injected in the body. We also want the lowest toxicity possible, because we want to be able to do longitudinal studies and also access fragile population or diseases. And of course, we want to develop tracers that are feasible for clinical translation, which are potential, which can bind to the target in human, but also in animal investigation. Okay, this next slide is a collection from a recent review of proposed PET tracer for imaging CB cannabinoid receptors 1. Has to be said that endogenous CB1 receptor ligands have moderate affinity and are highly lipophilic, so which means that they are not specific, only specific. So synthetic CB1 antagonists have been developed over the year. This is a collection of the most successful one from recent reviews. I would like to stress a couple of them, which according to the authors are the most promising. This is the first one, is based on carbon, and as you see, its efficacy has been proved in rodents, also in non-human primates, up to human, and also in pathologies, and also it has quite a good brain uptake, as is reported in this table. The second one is based on fluorine, it's more recent, and again, it has been its efficacy has been improved in rodents and human primates and humans and in diseases. It has even better values for the brain uptake. But I remind you, for a full list, I remind you to this very good review. While PET tracer for CB1 receptor are relatively easy to formulate, it's a bit more complicated for CB2 receptors. Actually, CB2 receptor expression was believed to be limited in healthy brain, although recent evidence kind of disproved this. However, what is interesting about CB2R is that they are upregulated on activated microglia cells, which makes PET imaging of the CB2 receptor an ideal target for imaging inflammation. Synthesizing CB2R tracers is not easy because the CB2 receptor are highly because of the high lipophilicity, the metabolic instability, unsuitable pharmacokinetics, and also lack of selectivity of the ligands over CB1, the receptor of type 1. Also, another thing that is hampering this search is the fact that there is a lack of selective CB2R antibody to confirm the findings using postmortem techniques like histology. However, recently CB2R crystal structure has been resolved, so there is a hope in the field that this will impulse the formulation of new tracers. This is a collection of the proposed one from the same reviews, with most of them have been tried on rodents, only rodents, or in spinal cord tissue, and they have also a modest brain uptake. Some of them are promising. Okay, so this last slide about PET tracer is looking at PET tracer for hydrolysis, especially FAAH and MAGL. This is important because the drugs that act as agonists or antagonists of cannabinoid receptors can cause, it's well known that they can cause dependence or addiction. So this is an alternate target, the enzyme that metabolize the neurotransmitters, EIE and 2-AG, and these are FAAH and MAGL. For both enzymes, both reversible and irreversible tracers have been proposed. The reversible ones are preferred, and not preferred because they are less dependent on perfusion and more on the actual density or activity of biological targets. However, especially regards FAAH, there was a major clinical trial incident related to an FAAH inhibitor, which slowed down the development, actually almost stopped the development of this kind of tracer, so it's currently an unmet clinical need. Again, this is from the same review, a list of the main PET tracer that have been proposed in the literature addressing these two enzymes. As you can see, especially for both of them, the subject and student in understudy are mostly rodents or non-human primates. There are really few experiments or proof of concept targeting humans. So the field is still pretty much under development. Okay, so that's it for now for a general overview about the PET technique, but I thought it would be nice to add also a few examples of the applications. This is the first paper of already a few years ago. This paper targets the availability of CB1 receptor after acute and chronic alcohol consumption and also in abstinence. Interestingly, the author find that while binge drinking causes an increase of the CB1R availability, the opposite effect is seen in chronic alcohol consumption, so up versus down regulation. And in chronic alcohol consumption, this reduction of CB1R availability is not recovered during early abstinence. This is a bit in line with this recent review where the authors compare different clinical studies addressing PET tracer for CB1 receptor in the context of alcohol use disorder, and they also found this difference pattern when comparing binge drinking versus chronic alcohol consumption. Another work that I would like to talk about is the use of CB1 receptor, the mapping of CB1 receptor in schizophrenia, which is really prominent mental condition. From this recent review, the authors highlighted the fact that while it seems that CB1R have a role in schizophrenia, this role is not clear because depending on the studies, depending on the study, both increase or decrease in schizophrenia of this kind of receptor image through PET have been reported. Notably, this is the work of Ceccarini, where an increase in CB1R binding in schizophrenia, both treated and free and drug-free groups compared to control, is highlighted. While in another work, the opposite pattern is found. So reduce CB1R availability in schizophrenia patient compared to control. In this case, you can see in the paper by Ranganathan at the right, you can see that this is something that you can find often in PET papers, PET results, that while PET used for the specificity of the molecular target, also an MRI image for anatomical reference is acquired. We will talk a little bit more about this. Because they can be acquired at the same time if the machine is a PET MRI, or they can be acquired in separate moments and then co-registered through co-registration techniques. But MRI is normally a good complement for PET imaging because PET has a much lower resolution compared to MRI. The authors of the review, Navarrete and co-workers, highlighted the role of confounding factors in this literature search. For example, symptom severity, sex, age, the use of these different PET tracers, different statistical methods, or other comorbidities that perhaps have affects this inhomogeneous trend in the result. So the author concludes that CB1R has indeed an important but yet complex and poorly understood role in schizophrenia. And another paper that I would like to discuss in this class is this one, kind of 10 years ago, but it's addressing CB1 receptors in Parkinson, using the fluorine-based tracer. And again, in this case, also there are inhomogeneity of the CB1R availability across the brain. In fact, in this paper, the authors highlight that the Parkinson patients showed a decreased CB1R availability in the substantial nigra when compared to control, but indeed the CB1R availability was relatively increased in other areas like the neuroskeletal, mesolimbic, mesocortical, dopaminergic projection areas. Let's move now to the second imaging technique that I'm going to talk about, which is SPECT, single photon emission tomography. Again, I will start with a brief overview. Single photon emission tomography is a nuclear imaging technique which is based on injection of radioactive tracer, a radiopharmaceutical, emitting single photons, gamma ray, during its decay process. So it's another kind of radioactive decay. The photons that are emitted are detected by a gamma camera surrounding the patient and are used to produce 3D images. So, as you can see, the rationale of SPECT imaging is quite similar to PET, and also in the context of the endocannabinoid system, so injecting radioligands binding to the cannabinoid receptor. What is different is the fact that most of the tracers are based on the photon-emitting iodine 123, which is a compound which has optimal properties for labeling low molecular weight organic compounds while retaining biological activity. So again, there is the tracer is constituted by a part that is binding to the target of interest, and another part which is radioactive, and in this case is emitting photons. And again, this is used to produce 3D maps of the distribution of the target of interest. In this case, I think this was one of the first trials in vivo of SPECT tracer, paper from 2004. I think immediately, and again, this is accompanied by MRI images for the anatomical localization, but you can already see that the resolution of the SPECT is quite low even compared with PET, and of course much lower than MRI. Which is the physical principle behind the SPECT? Iodine 123 decays through a process that is named electron capture. Electron capture is the radioactive decay process by which an atom's inner orbital electron is absorbed within the nucleus, followed by the conversion of a proton to a neutron and an emission of a neutrino, as schematized below. The resulting daughter nuclide, it's normally in an excited state, and so it transitions to its ground state. In this transition, a gamma ray is emitted. In the case of iodine 123, the decay of the parent nuclei results in the daughter, tellurium 123, in an excited state, so it releases the energy of a 159 kilo electron volt in excess through a light photon. The tracer, again, so this is again the experimental flow. You can see that it's pretty similar to the PET imaging. The tracer is composed by a biologically active molecule and a photon-emitting atom. Most of the cases, iodine 123. Again, it gets produced locally close to the experimental site, and then it's injected in the body through injection. And the tracer binds to the target, receptor in this case, and it starts to go radioactive decay through this process of electron capture, emitting gamma rays, which are then detected by a camera, which is it's perhaps simpler than a PET scan. It's just a rotating camera that rotates around the body of the patient. A few details about this camera: it consists of a collimator, which allows only gamma rays traveling in a specific direction to pass through, and a scintillation crystal that converts these gamma ray photons into visible light. And then these emitted photons are detected by an array of photomultiplier tubes and converted into electrical signal. This is what is detected by the detector. The image, the 3D image is done offline by reconstructing all these 3D projection of the rotating camera, which are projections of the 3D distribution of the tracers. And in this case, the maps represent the counts or counts per pixel, which is the number of detected events. So this is the unit of measurements. A few words about where these tracers are produced is device which is called the cyclotron. It's a device in which charged particles like protons, deuterons, and alpha particles can be accelerated to very high speed in a circular path and can be directed on the target, which normally consists of stable isotopes, and then converted into corresponding radionuclides by nuclear reactions. This is a scheme that you will see below. The result is radioactive atoms that can be separated from the target material and radiochemically processed to make the tracers. And this is a real-life picture. I think this is a GE cyclotron. So you can see that the dimensions are relatively compact. And it has to be, as I said, it has to be in the proximity of the PET or the SPECT scanner. A few words on SPECT applications, although there are far less applications compared to PET imaging. In this study, this is this is the one that I showed before in the pictures. It's a study from 2004 in biological psychiatry, and is the first proof of concept of the use of a tracer, of a SPECT tracer, binding to CB1 receptors in patients of Tourette syndrome. So it was the first time it was applied in vivo. This is the tracer. It's based on, as I said, almost all of them are based in U923. So in this paper, the authors quantified the distribution of the tracer using kinetic modeling. In the upper plot, there is the activity curve over time in the plasma. And in the lower graph, there is the measured T-cell response in one area of the brain, which we're specifically interested in, which is the nuclear lentiform, and also the occipital region for reference. You probably can see, well, there is an arrow, but you probably can see a moderate peak of uptake in the lentiform nuclei, which is where the arrow is, around 1 hour post-injection. Again, it's quite clear that the resolution of the SPECT image is quite poor compared to the PET, but has to be said, although I will talk about it in details later, that SPECT has much higher availability in hospitals and it's more affordable. So this is why sometimes it can be preferred over PET. Okay, so let's go to the third part of this class, which targets MRI technique. While in a way PET and SPECT can provide kind of a direct quantification or imaging of molecular targets relevant in the endocannabinoid system, MRI provides much higher resolution, but a more indirect quantification of activity changes following alteration in the endocannabinoid system or structural changes. We will see the details. But of course, it has the advantage of having much higher resolution. Okay, well, again, let's start with an overview. MRI is an imaging technique that exploits the magnetic properties of water in the tissue to extract information about macro, micro, and structure and function. So instead of injecting a tracer, the tracer, in a way, is the water that is already in the tissue. This means there is much higher signal, but that, of course, specificity is more challenging to achieve. As I said briefly before, MR-based imaging of the endocannabinoid system can be used to quantify several aspects of the system structure and function following changes or interaction with the endocannabinoid system. So, for example, the most used techniques is functional MRI, which is used to detect changes in functional connectivity associated with the endocannabinoid system activity. Another technique that is pretty used is due to the role of the endocannabinoid receptor in regulating the dynamic of the blood, the blood flow and dynamics, is the sequences based on imaging cerebral blood flow associated with endocannabinoid system activity. I also talk a little bit in this class about spectroscopy, although it's not strictly an imaging technique, it's a spectroscopy technique, but it's in the MRI, the anatomical image can be used to localize an area of interest where the spectroscopy analysis is performed, which allows to extract concentration, relative concentration of metabolites, which are implied in the endocannabinoid system in specific parts of the brain. And finally, perhaps the more indirect probe is the methods based in structural MRI, both micro and microstructure, so different that can allow to image and quantify different in various aspects of brain microstructure. Okay, so let's talk a little bit about the physical principle. MRI is, compared to PET and SPECT, is more complex technique, but it's even more versatile. Different contrasts, there are a number of different contrasts depending on what is the challenge of interest in the tissue. And the contrast, the idea behind MRI is based on the fact that water is everywhere in the body, that water is composed by 2 atoms of hydrogen, 1 atom of oxygen. In the context of MRI, what is important are the hydrogen atoms, because they possess magnetic properties, specific magnetic properties, which are modeled or resumed by the concept of spin, which is the intrinsic angular moment associated with the constituent atoms of the molecule in their arrangement. The spin, normally they are represented by this arrow that is imaged, although this is not what happened, but imaged as an axis of rotation for the molecule. This is not what happened. There is no actual rotation. It's just an intrinsic angular momentum. However, this schematization can be useful to visualize the fact that the spins are oriented randomly in normal life. They are oriented randomly, but in presence of a strong external magnetic field, they can align to the direction of the magnetic field. So this is the first thing. The spin of the water contained in the body, in the brain, in the body, can be aligned by a constant intense magnetic field. And also, if we superimpose to this constant magnetic field a smaller one, another one with less intensity but oscillatory, they can actually be manipulated and rotated. They can be manipulated, bringing them into a position out of their equilibrium, and the way in which they go back to the position of the equilibrium, they do this by releasing energy. And this energy is, the way in which they release this energy, the intensity of this energy, depends on mostly 2 factors: the spin position and the environment of the spin. So, by coding the position of the origin of the signal, it is possible to segregate the signal coming from each part of the brain, so basically reconstruct a 3D image, a 3D map of the brain. These maps will in turn have display different contrasts depending on the different tissues, because different environment, for example, gray matter, white matter, CSF, makes the spin relax back to the position of equilibrium in a different way. So this is why MRI is used to image, to photograph in a way, contrast between soft tissues, different adjacent soft tissues, as you can see in this kind of maps. But actually there are 4 different kinds of contrasts here, because I can change the kind of stimulation, the oscillatory field that I impart to the sample. So by manipulating this, by changing this, I can have different contrasts. In this case, I think it's a contrast based on different relaxation times, based on photon density. I can make contrast based on diffusion, I will see later. So this is why MRI is extremely versatile as a technique. And actually there is a whole field of research which is just developing new stimulation protocols, which are called pulse sequences, because of this oscillatory property, which is to develop these pulse sequences, accessing newer and more advanced and more specific aspects of the structure or microstructure. One very, very, very interesting and very relevant contrast that can be accessed is the contrast that is recorded in the brain, where there is a flow of oxygenated blood that is related to that area of the brain being active. So this is the key behind the technique called functional magnetic resonance imaging, or functional MRI or fMRI. As neural activity increases, the local blood flow increases too, supplying more oxygenated blood to the active area. Which means that the ratio between deoxyhemoglobin and hemoglobin changes. Deoxyhemoglobin and oxyhemoglobin have very different magnetic properties. The first one is paramagnetic, and the second one is diamagnetic. They have a different impact in the way in which the spin of the water go back to the equilibrium. Specifically, in presence of paramagnetic compounds, the going back to the equilibrium is called, in this case, the contrast is T2 star, it's relaxation time for the spin to get back to the equilibrium, is faster, while in presence of oxyhemoglobin becomes slower. So again, activation of a specific brain area changes the balancing between oxy and the oxyhemoglobin, and it changes the curves of relaxation of the signal intensity versus time. In red, the curve that will correspond to an area or a moment of time in which there is an activation of that area compared to baseline. So this is all is due to the changes in the concentration between oxy and the oxyhemoglobin. So this effect is called the BOLT, or blood oxygen level dependent effect, and is the contrast that is at the basis of functional MRI. So what is an fMRI experiment? It's basically acquiring this kind of T2 weighted images densely over time. Normally the repetition time is 1 to 2 seconds, and by repeatedly acquiring them over time, fMRI can create series of images of fluctuation of brain activities. These areas can be analyzed, and these images can be analyzed and interpreted according to the kind of the experiment. Task-based fMRI can detect areas which are activated or inactivated during a task. So in this case, the time series will be correlated with the task, the features of the task. But another branch, very interesting, is just looking at the intrinsic activity of the system when it's resting, and it's also called rest-sustained connectivity. There are specific patterns and a specific network that gets activated when the brain is at rest. This is very consistent across subjects, across species, so this is another field of a lot of interest. The idea that is behind fMRI is always that correlation between fluctuation of the signal in different areas indicates connectivity. So this is why fMRI is used to look at brain connectivity. This is of course an assumption that has some limitation, but as I said, in general, rest-sustained networks are very robust across subjects, across repetition, and across species. So digging a little bit more into the concept of connectivity, as I said, the connectivity is basically a correlation between time series from different locations in the brain. So in this case, I think it's the caudate, left and right. One can calculate the correlation between the two time series. This signal is basically the intensity of the MRI signal measured across time. By calculating the correlation, the pairwise correlation of all the regions in the brain, one can construct a matrix of functional connectivity. In this case, the correlation is probably very high because it's the same region but in the two different hemispheres. But if I do this for all ROI versus all ROI, I can build this kind of matrix in which I can distinguish. I can actually even threshold, just setting a threshold of what is the significant connectivity, and just see areas that are connected versus areas that are not connected. Okay, so this is a matrix of functional connectivity, and there are tools that have been developed in the framework of the graph theory that are specifically looking at this kind of mathematical objects. And these tools are very useful because they can basically find, in this matrix connectivity, they can find important hubs that are important for the system, nodes within the hubs that are important, hubs that act as a connector. So there are all a number of measures, and I invite you to look at this paper, to look at the different measures that can be extracted using graph theory. And eventually this matrix can also be visualized by the different nodes and connection, where of course all the connection, the presence of the connection indicates significant correlation between the time series, and the strength means the intensity of this correlation. So this is a whole area of research, and I remind you to this bibliography too if you want to know more. As I said, I would like also to mention something about MR-based spectroscopy, because though it's not strictly an imaging technique, I think it's very useful, can be very useful, and especially and also in the context of the endocannabinoid system. So the idea behind the MRI spectroscopy is that the resonance frequency, the frequency at which the nuclei absorb the energy in an MRI experiment of a given nucleus, is influenced by its chemical environment. So that different chemical compounds or molecular structure have different resonance frequency. And this is called the phenomenon of the chemical shift. So what do I do in an MRI spectroscopy, in an MRI spectroscopy experiment? I select a volume of interest for my stimulation, and then I map the different frequency in this volume of interest. These frequencies will appear as peaks in the spectrum, and will correspond to different chemicals. And this is an example from a recent review. Then, with the spectra, by comparing this spectrum with a spectral pattern of reference, I can estimate the concentration of various metabolites of interest. In this case, the one that is shown in the picture, the glutamate, creatine, choline, myoinositol, etc. So all metabolites of interest. And of course, it can be used also to quantify metabolites that are important in the endocannabinoid system. Another technique that I would like to present is the arterial spin label. Arterial spin labeling is used to measure the cerebral blood flow non-invasively. It's a very powerful tool, and it's based on the fact that I can label an area of the body in which there is blood that will later go into the brain. So a specific region of the arterial blood flowing into the brain is labeled magnetically through a pulse, by applying a frequency pulse to selectively invert the mineralization of the arterial blood protons. After this labeling, the labeled blood will travel to the brain tissue, normally very quickly. And what happens is that the labeled blood mixes with a non-labeled blood that is already in the brain capillaries, resulting in a decrease of the mineralization of the labeled blood. By acquiring two images, one with the arterial blood labeled and another without, which is the control image, one can do the subtraction of this, and the subtraction is actually proportional to the labeled blood perfusion, proportional to the cerebral blood flow. If this is it's calibrated by this different image is calibrated, CBF values can be extracted, quantified in units of milliliter per 100 gram per minute. And these are maps of the cerebral blood flow. And this is very interesting again because it can be quantified non-invasively. Okay, the last part of MRI techniques, as you see, there are it's a much richer technique compared to PET and SPECT, because there are a number of different kind of aspects of the tissue structure function that can be analyzed. Is the diffusion based? What is diffusion? Well, diffusion is the random motion of all particles which have an absolute temperature bigger than 0, and they can visualize. They can be tracked. Diffusion can be tracked by diffusion-weighted MRI because there are sequences that allow to track the displacement of particles as if we were putting a red tag on water molecules and observing how they displace. Diffusion in this picture can be visualized as an ink, a colored drop of ink in water. As you can see, this random Brownian motion of all the particles happens in normal life, because if you drop colored ink into the water, the ink will start to diffuse in the recipient until it has completely filled it. This can be visualized by diffusion MRI, by this way of tagging. And why is this important? Because by tracking how the particle displays in this environment, it's possible to indirectly measure the microstructural properties of such environment. For example, if the water molecules are diffusing in a geometry which is made of cylinders, it's much more likely that it will diffuse in the direction of the cylinder compared to the orthogonal directions. This is actually a very good model for diffusion in gray white matter, which is under a number of assumptions made of axons, bunch of axons, myelinated axons, and where the water can diffuse more easily in the axon direction. Mathematically, this diffusion is described as with a structure that is called a tensor, and this tensor is the easiest mathematical structure that is able to capture this. The word is anisotropy, the fact that the diffusion is facilitated along one axis. And this was discovered already kind of 30 years ago, and since then it has been applied a lot to investigate white matter structure. From this tensor, two kind of maps are extracted. One is the fractional anisotropy, which is basically the amount how anisotropic is the diffusion, so which is the difference between the principal axis of diffusion, in this case is called lambda 1, compared to the other two, lambda 2 and lambda 3. And when you see white in the map, this is areas of highly packed white matter fibers, where the diffusion is really anisotropic. Another marker is of the map is the mean diffusivity, which is the average of the diffusivity along the three axes. Both of them are related to microstructural indexes of microstructural integrity. Mathematically, the fractional anisotropy is the variance of the diffusion across a different direction. And what is important is that it's high in regions when the diffusion is most directional, and for this reason it relates to the integrity of white matter fiber bundles. This is an example of how the tensor describing the diffusion could be in areas of a high fractional anisotropy and low fractional anisotropy. Conversely, the mean diffusivity is the mean of the diffusion coefficient across all directions. It's not sensitive to anisotropy. Is again a correlation of tissue integrity. For example, is altered in acute and chronic stroke, even in another number of diseases. And this is how high versus low mean diffusivity would look like. By looking at the direction of the principal diffusivity, we say that this is facilitated along the fiber tracks. If I visualize it in the brain according to a color code where anterior posterior orientations are green, left right are red, and superior inferior are blue, I can create this kind of color coded map. But what is interesting is that if I then look at the directions at the orientation of the principal axis of diffusion in each voxel or element of the image, and then I reconstruct the trajectories of white matter by displacing from one voxel to another, just following this principal orientation of diffusion, I can do so, and by doing so, I can reconstruct the fiber tracks from every point of the brain to every point of the brain. So eventually, what it will look like is something like this. This is a collection of all the fibers, of white matter fibers within the brain, colored according to the direction of the color coding. So red is left right, blue is superior inferior, and green is anterior posterior. Okay, so let's dig a little bit into some MRI applications. So the first couple of papers that I'm presenting look at the effect of cannabis use on brain function. So in this case, the author showed that cannabis users had higher susceptibility to memory illusion, and this is similar what is observed in certain neurological and psychiatric population and also in elderly individuals. But they also further, through using fMRI, they further identified the functional substrate of this deficit, and they identified it as the hypoactivation of a series of spatially distributed brain regions participating in the network which is involved in semantic and episodic retrieval. They also find an association between the false memory rejection and the self-reported use of cannabis lifetime, meaning that suggesting that this effect is also quantitative. Another paper going in a similar direction, the effect of cannabis use on brain function, is this fMRI study, in which the acute effect of the tetrahydrocannabinoid, THC, which is an endocannabinoid agonist, on brain function is quantified in relation to the emotional processing. To do this, the author measured the performance and reactivity during matching of stimuli with a negative or a positive content, fearful or happy faces, after placebo or THC administration. So they highlighted that they found that this task activated a network of brain regions which are called amygdala, orbitofrontal gyrus, hippocampus, parietal gyrus, prefrontal cortex, and regions in the occipital cortex. And that, interestingly, THC use interacted with emotional content as activity in this network was reduced for negative content, but while it was increased for positive content. On the same line, by looking at the difference in structure, I would like to discuss this quite recent paper about THC exposure and looking at microstructure of the cortex in amygdala of young adults. So the authors found significant association between THC exposure and microstructural parameters in brain areas that are related to silence processing, emotional regulation, and decision making. And again, this effect was increased for the more the dose-dependent the dose which was self-reported in cannabis usage. Specifically, the parameters that they measure in the study with structural MRI, microstructural MRI, is the neurite dispersion. And what they found is that by comparing controls and cannabis users, they found an increased dispersion in the neurites, which are the pathophysiological underpinning of this observed contrast. It's not clear, cannot be clear, but just an MRI study. It may relate to dendrites, but it may also relate to processes of the microglia. Okay, this is another quite recent paper, which is again on the effect of cannabis use, but on the cerebral blood flow. In this case, CBD increases cerebral blood flow to key regions involved in memory processing, particularly the hippocampus. So this is comparing placebo versus cannabidiol, acute cannabidiol administration. And the authors speculate that this is a potential mechanism for CBD for a range can be useful in a range of conditions associated with altered memory processing like Alzheimer's disease, schizophrenia, PTSD, and cannabis use disorder. This other paper is also quite recent, and this is looking at young adults with and without psychotic disorders. In this case, they used PET to quantify FAH activity. They also used MRI spectroscopy to look at glutamate and glutamine in the hippocampus. And then they also looked at, they used also MRI, conventional MRI for localization and for high structural resolution. What they found is in a series of regions relevant for memory, they found a correlation between the regional FAH activity measured by SPECT and the concentration of the hippocampus glutamate and glutamate in a number of regions. This other paper that I would like to present is another combined PET and fMRI study looking at FAH enzyme and amygdala connectivity. The author found the upregulation of the endocannabinoid enzyme FAH has been linked to abnormal activity in the frontoamygdala circuits, which is a hallmark of PTSD, post-traumatic disorders. What they found through fMRI analysis and PET is that the FFH levels in the amygdala were negatively correlated with functional connectivity between the amygdala and the prefrontal cortex. And what is interesting about this paper is that they used a targeted approach by looking at the region of interest, connectivity region of interest, but they also used C2 box cell analysis, in which only the starting seed for the connectivity is chosen, and then the time series is compared with every other box in the brain, and this is a more unbiased estimation, and they confirmed the same correlation between the connectivity with the prefrontal cortex, areas subserved with stress and affect control. Okay, so let's move forward for a little bit of a comparison across different methods. I've talked a little bit already about the combined use of PET MRI. It's a hybrid imaging technology that incorporates the two techniques, MRI and PET, into the same machine. So it's apparently, this is the one from GE, I think, and it's apparently similar to a regular MRI, but it has double functionality. In this case, this is a work of looking at glucose metabolism with PET. This is the first image in the plot in the picture, and this is combined. This is the typical low resolution of PET, but this is combined with high resolution MRI, so you can have diffused contrast. Which is the caveat that the performance of the machine, the resulting quality of the image of the machine doing both kind of analysis can be worse than single modalities. So it's not always the best choice to use a PET MRI scanner, but sometimes it can be interesting to acquire the two modalities in two different moments, just by exploiting the best performance for the hardware, and then co-localize the two images by means of registration. Let's move now to a more quantitative comparison between MRI and PET or SPECT. So MRI, as you can see also in this image, has a very high spatial resolution, 1 to millimeters. You can go even lower with modern scanners. It uses intrinsic contrast water in the tissue, so it doesn't use ionizing radiation. It offers the possibility to achieve multi-contrast imaging, functional, structural, blood flow, spectroscopy, etc., and also has this access to the functionality. Which are the downsides of using MRI? It's generally pretty long. An MRI scan is pretty long compared to a PET, and it's also expensive. The contraindication for undergoing an MRI examination is having metal in the body. So, for example, pacemaker is not allowed. There have been also a few accidents with metals in the room. And then probably the biggest contraindication or limitation is that it's the specificity is limited, definitely limited compared to specificity of SPECT. On the other hand, PET again the specificity, so it can do molecular imaging. PET is faster, it's dynamic, it allows the acquisition of the dynamic images, capturing the changes in the uptake and distribution of tracers or enzymes over time. The downsides are the low resolution. We are talking about 3 to 6 millimeters. The radiation exposure, which makes it not suitable for repeated longitudinal examination, not suitable in case of specific fragile population. And the costs are high, well, probably not as high as MRI, but still high between the scanner and the cyclotron. And also there may be limited malleability, especially because the examination setup must have both the scanner and the cyclotron to produce or in the proximity to produce the tracers. And finally, the comparison between PET and SPECT. I already said a few things, but in this, I found this paper which is quite interesting because it's looking at the same molecular target but with a tracer based on SPECT and a tracer based on PET. So in this case, the resolution has been matched, relatively matched. It's a bit higher for PET. This is the mouse brain, but to achieve similar contrast, the injected radioactivity was 10 times higher for SPECT. And this is demonstrating the significant difference in detection sensitivity between the two techniques. So, although they have similar mechanisms of action, radioligand, the radioligand change, photon versus electron emission, they have very different spatial resolution, 6 millimeters versus 12 millimeters of SPECT. Higher sensitivity, PET has higher sensitivity, can detect lower concentration of radio tracers, but SPECT is more affordable. It's more, there are more sites in the world which have SPECT, and it's cheaper. So it's always a balance of the two techniques. Okay, my class is finished. I hope you enjoyed it, and I just want to quickly acknowledge my laboratory and my collaborators. And if you have questions, don't hesitate to drop me an email to this address, and I will be very happy to answer. Thank you very much.